were able to inhibit mast cells. And in that study, the trigger of the mast cells was a substance P, a neuropeptide that is found in many places in the body. And just to convince you that it can stimulate the mast cells, you can see a very nice dose response with increasing dosage of substance P and release of histamine. So how does this tie to chronic fatigue? Well, we took a little blood from some patients that we had access to, and we measured levels of substance P in the blood. And as was reported earlier for peptide Y, we actually found that substance P was actually higher in these patients, and we're desperately trying to find, and I've spoken to some of you, more material to study actually substance P in addition to what I'll be telling you in a few minutes. So, what are mast cells? Well, we always thought that they were involved only in allergic reactions and that they're stimulated by Ig binding to specific receptors and causing degranulation with histamine coming out. Well, over the last few years, both we and others have shown that the mast cells can also release a number of cytokines, growth factors, neuropeptides without ever degranulating. So you'll never see them changing actually in plain histology. And in fact, there are numerous triggers of the mast cells and about 50 different mediators that come out of the mast cell, whether the cell degranulates or not. So we used to think that they're involved in allergy and in itching, but as it turns out, practically every course in pathology now that I know of now starts by invoking release from the mast cell so that there is vasodilation, so that there is vasoattraction of various immune cells, and also expression of endothelial adhesion molecules that allow the leukocytes to come out. So in numerous studies, when inflammation was trying to be induced in mast cell deficient animals, it failed. So there's no question that mast cells are involved in inflammation. But what is fascinating to me, and I made mention to this yesterday, is a number of combination of triggers has actually augmenting or synergistic effects. Substance P and IL-33, a new cytokine, substance P and IL-9 that's been shown to be neurotoxic to glial cells, substance P and mercury, the list goes on. So as I said yesterday, I don't think there's one molecule that would be important, but actually a combination of molecules. And I tend to sort of agree with Ben that the action might be actually in the brain, but invoking actually molecules of the sort. Now, increasing evidence over the last few years has been indicating that the mast cells are actually involved, although to different extents, in a number of diseases. I'll try to talk a little bit about chronic fatigue, but clearly, not only they're involved in a number of diseases, not only they don't degranulate in these diseases, except maybe for allergies and asthma, but many of these diseases tend to be comorbid, which indicates to me that there's something underlying that might have a similar background. And because of that, the American Academy, along with the Mastocytosis Society, put out a video that is actually available to all physicians and others in the States. They didn't call it allergic symptoms. They called it mast cell activation symptomatology, because exactly that, mast cells could be involved in a number of diseases. And as Chris sort of alluded to, we've known each other because I've been involved in interstitial cystitis, where we actually see bladder mastocytosis in many patients. Now, why brain, why chronic fatigue? Mast cells are found very close to nerve endings. This is actually from the dura of a rat, next to a blood vessel. And if you magnify it about 200,000 times, you see part of a mast cell, part of another mast cell, and this is actually a synaptic terminus with synaptic vesicles. So mast cells are very close to nerves, very close to neurons. Here you actually see a cross-section of a blood vessel, a pericyte endothelial cell. They make up the blood-brain barrier. The mast cell is actually hugging this. And in fact, this particular picture was taken, and it was on the cover of Brain Research Review some years ago when we wrote a review about mast cells in the brain. Amazingly, you have as many mast cells in your diencephalon as you have in your skin. The brain never gets allergic reactions. So there have got to be other triggers that trigger these cells. So we became very interested for the mitochondria for three reasons. They make energy. We need energy. They actually buffer calcium ions. You need calcium ions for secretion. And to the extent that you have fatigue, we thought maybe there's something there, even though we heard yesterday there's nothing obvious, at least in the muscle. What is fascinating, if you stain mitochondria with mitotracker, that stains mitochondria are red, you see that their distribution in numbers varies. For instance, epithelial cells have tons of them. This is the nucleus. Kidney cells have fewer, and mast cells have even fewer, and this is the nucleus, or it is the impression of the nucleus. So, going into the results very quickly, if you stain mast cells, these are human umbilical cord derived normal mast cells. They take about 8 weeks to grow. You can actually see that the mast cells concentrate around the nucleus. Here is a Nomarski optics of a mast cell pretty normal. This is the nucleus. And you can see here also that the mitochondria are very close to the nucleus. If you stimulate the cells with substance P, you see the mitochondria start spreading around. You can see the mast cell started bubbling as it sort of degranulates, and now you can see the mitochondria emerge spreading around. So you can also do this by staining the mast cells with something called lysotracker that stains the granules because they have low pH, about 5.5. So now you can see actually, by doing the two, that the mitochondria start spreading around, and if you do actually quantitation, you can see in controls they're kind of close to the nucleus here, and here they actually spread out, and you can see image analysis, and you can show actually that there is movement of the mitochondria closer to the cell surface. And if you stimulate the cells and you look at them with electron microscopy, you can see here's the nucleus part of a cell. You see large mitochondria very close to the nucleus. You stimulate the cells. These are exocytotic pits. This material is being secreted from the granules. Now you see the mitochondria very close to the cell surface, and they're much smaller, and you can actually show that with image analysis as well. So the summary from this was the mitochondrial fission and translocation occurs along with substance P-induced degranulation, and I'm not showing you to now, but different triggers can do that, and not just substance P, but in other different neuropeptides such as neurotensin and NPY will do the same. But the second hypothesis was, is this mandatory for the cells to release? We know from many other studies that a protein called DRP1 is absolutely necessary, and upon being its phosphorylation, goes to the mitochondria and actually allows the mitochondria to undergo fission. So we decided basically to block this molecule, DRP1, and see what happens. And in fact, here is control. This little circle indicates where the mitochondria, the red, are. If you actually stimulate the cells, as I showed you before, the mitochondria go all over the place. But if you block actually DRP1, the mitochondria stay exactly where they were, and in fact, we inhibit secretion. And if we actually express or knock down DRP1 in human cells, you can see actually we can drop down expression, and we drop down secretion from mast cells as well. So the summary from this is mitochondrial fission and translocation are actually required for mast cell activation, and it's not just a phenomenon that occurs just because. The third hypothesis, which was actually more appealing and sort of fascinating to us, is could it be the mitochondrial DNA and other components are secreted extracellularly as the mitochondria undergo fission and move to the cell surface? And in fact, early on you could see here, for instance, cells stimulated by substance P. You can see actually particles that are stained with mitotracker, and some are in the blocks. They would not stain unless they were intact. Mitotracker stains only intact mitochondria or portions of mitochondria. But then what we did is we took the supernatant from mast cells that were activated with substance P, and we looked for two DNA species, 7S and Cyto B, which are mitochondrial specific. And you can see as we increase substance P concentration, you can see tons of mitochondrial DNA in the supernatant fluid. We looked for GDPH, we would indicate nuclear DNA. There was absolutely nothing there. And in fact, different techniques to show that the cells were alive. There was no necrosis, no apoptosis. So this was extracellular free-floating DNA that was being released. So then the question is, if you have DNA being released, could it actually stimulate other cells? Because remember, mitochondria were actually pathogens that became symbiotic over millions of years, and in fact, they never leave the cell. They're destroyed inside the cell by autophagy. So if they leave the cell, they will be mistaken by the body basically as a pathogen, and they will mount an immune response. Make a long story short, we actually isolated mitochondria from sarcoma cells that we actually cultured. You can put them on neutrophils that put IL-8. I'll show you these results. You can put them on neurons, it causes neurotoxicity. You can put them on glial cells, activates the hell out of glial cells through TLR receptors. Let me just show you what happens on the mast cells, which means that this mitochondria that is released has both autocrine and paracrine effects. So here you can see increasing concentrations of mitochondria. These are, again, cultured human normal mast cells, and you can see TNF production and IL-8 production as we increase the amount of mitochondria, and here is substance P for comparison. So the summary from this is mitochondria and mitochondrial DNA are released from activated mast cells, that intact and sonicated mitochondria stimulate mast cell degranulation, so it might be more than just mitochondrial DNA that is being released. and sonicated mitochondria stimulate mast cells to release TNF and IL-8, which means that we're releasing something from inside intact mitochondria, which not only might be mitochondrial DNA, but we have a lot of evidence that ATP that is released from these mitochondria can also stimulate the cells. So as a summary, and I'm getting close to the end, we can activate the mast cells. We know now mitochondria have to undergo fission and move to close to the cell surface. There is release without degranulation of some cytokines, but there is actually TNF released along with mitochondrial DNA. And I have to stress, the mast cells are the only immune cells in the body that store TNF in granules. Every other cell type makes TNF from scratch, just like other cytokines are made from scratch. That takes 24 to 48 hours.